So yeah, basically Waldenström's has two basic genetic features. This is MYD88 and CXCR4. So obviously all of the studies focus on these two genes. And I think this is the first study that actually, on a single cell level, you can compare patients which have the MYD88 mutated phenotype, which represents about 95% of our patients, and compare them to the 5% of the non-mutated wild-type MYD88 patients. Obviously, these patients have a distinct clinical phenotype. They don't do that well with ibrutinib therapy. They have adversely affected overall survival. So we want to see at the single cell level, which you actually can pick out really rare population, what happens and what are we missing out, so we can actually target more sensitively. MYD88 is used to actually differentially diagnose Waldenström, but obviously there are other lymphomas such as MZL or LPLs that have this mutation, and it's hard to actually distinguish the differences. So you really need these tools. Obviously, MYD88 CXCR4 are really strong genes to use them for diagnosis, but we need more genes and we need more markers to actually distinguish between different lymphomas. So single cell, I think, is one promising, well, not diagnostic tool, but obviously it's a promising tool where it can lead us to actually find which are the important genes that might be differentially expressed, and then we can go more targeted and use this in more diagnostic tools for predicting much better patients with Waldenström's.